English somehow. I hate English. I like having.
George, are you like, do you really have anything going on?
Yeah, Miss Carambus. Yeah, no, I just hang her off. Miss Carambus.
Yeah, you don't have something.
Yeah, no, she will accept bribes.
Okay, all right, thank you.
They are from us.
Thank you. Yeah, picking up where we left off from our last class, we were looking at the different R groups, okay, as our activity and annotating our data books. Yeah, so looking at acidic side group, so those that have got a carboxyl functional group in the R group. So we're not talking about the carboxyl group as part of the base part of the amino acid. No, sorry, the acid part of the amino acid, right? Talking about the R group itself. So there's only two, right? And we've got the convenience of the fact that it says acid in its name. Got our base functional groups. I'm not entirely sure about histidine and tryptophan because it's not technically an NH2, but it is an NH. But it will be slightly basic, yeah. But the main basic groups are those with a primary amine, right? We can consider histidine and tryptophan more so being just a polar functional group. Okay, those that have sulfur within the group, cysteine is the one in particular that we need to pay attention to, less so methionine. There's a reason for that. We'll talk about that a bit today. And then our polar functional group, so anything with something other than a carbon or a... Now, I would make the argument that methionine is not polar, and that's mainly because, right, there's not a lot of difference in electronegativity between the sulfur and carbon, right, in order for that to be a polarized bond. It will be ever so slightly polar, but that difference in electronegativity is less than 0.5. So methionine, even though it's got that sulfur present, sulfur's not really all that electronegative compared to oxygen within the same group. Okay. Non-polar, obviously, is going to be the other ones. We've got these rather bulky alkyl chains, or these large ring in the case of phenylalanine. Proline, right? Proline's really interesting, and I pointed that out last time. This is the R group pointing down, right? Unlike all the others that are pointing up in terms of their structure, proline, it's pointing down and it loops back on itself, right, to the amino functional group here. Okay. So this is where we got up to, the Zwitter ion, right? So polar nature of the amino and the carboxyl amino acids are soluble in water. Amino group can act as a base, right? Remember Lowry Bronsted from last year, acid-base reactions, that a base is a proton acceptor from the amino functional group, the carboxy group, right? The COOH acts as an acid as it will donate its proton. So any particular pH, the amino acid will be in, right? This is the average form called the Zwitter ion. So where the amino group has absorbed or has accepted the proton, and the acid part of the functional group, right, has donated the proton. So this is call it average, or it's the Zwitter ion, or also called the dipolar ion. Right, it's ion. Ion means a charged particle. We can see it's got both charged ends. Yeah, this is the important bit, right? So the pH affects how that functional group looks like. Yeah, so low pH, high acidity. Yeah, at high acidity, we've got the amino functional group absorbs that proton. Yeah. At a neutral pH, we've got the Zwitter ion in the middle. At high pH, right, we lose the proton off the COOH, and we just have our normal NH2. Does that make sense? Yeah? And the pH also affects, right, those acidic or basic functional groups. So if we can go back to here, at very high pH, both COOH functional groups will be deprotonated, yeah, in aspartic acid and glutamic acid. So both the COOH is part of the amino acid and that within the functional group will be deprotonated. Same thing at very low pHs, right? At very low pHs, the NH2 groups as arginine, asparagine, glutamine, and lysine, that amino functional group will also be protonated at very low pHs, right? Not just the NH2 as part of the main base part of the chain. That makes sense? Yeah? So pH affects the protonation or deprotonation of amino or carboxyl functional groups. Yeah? And that means that the bonding changes with respect to pH. We're going to talk about that today. All right. We've looked a bit at, we know what an amide is. We've looked at primary amides. Remember, we need to be able to recognize that. We don't need to name amides, but we need to recognize amides. So primary amides is when it's that we've got that condensation reaction between a primary amine and a carboxylic acid to give us an amide. Thanks, Anton. Yeah? Proteins, right? When I take the amine from one end of an amino acid and the carboxyl group, carboxyl end, undergoes condensation reaction. Condensation, water as a byproduct, forms the amide link, right? Also referred to as a peptide link. So amide is the functional group. Technically speaking, it's a secondary amide, yeah, given that I've got something other than H attached to the amine on this side. So the red and the blue here would refer to that CHR in the middle. Yeah, so that's going to change. Dipeptides, di means two, right? So that means two amino acids joined together and we get a peptide link or amide functional group. Now, order is incredibly important, right? So this is glycine and alanine joined together, right? But it's joined together in the order gly-ala. Whereas if I have ala-gly, right? So there are three-letter abbreviations. So gly-ala will look like gly-ala Right, so that's gly-ala is not the same as ala-gly. Right, do you see how they're different? Okay, so the order is important. Polypeptides, poly obviously means many, peptide link or amide link. Yeah, we can join all them together. Section of the polypeptide chain is shown highlighting the peptide link. The amino end is called the N-terminal. So this would be the N-terminal, and this is the C-terminal, right? Or the carboxyl end. So this section of a polypeptide would be referred to as val, cer, cis, ala, right? So we name our peptide chains with those by linking together those three-letter abbreviations of our amino acids. So order refers to what's called the primary structure, right? If we know this, if you've done DNA or if you've done proteins before, you should know what this is, right? So biologists in the room, this should be really straightforward. Primary structure of a protein is the order of amino acids. The bonding of the primary structure is covalent by those amide functional groups. So here's a chain of a protein, or protein really is just a polypeptide. Yeah, I can look at, so we call these the residues or the R groups, right? Amino acid residues is one way to think about it. I've got cysteine, CH2SH, arginine, it's got that functional group, whatever, valine, asparagine, isoleucine, cysteine, so blah, blah, blah. So the order of the amino acids gives us our primary structure of a protein. Remembering that proteins can be like thousands of amino acids joined together. Obviously, not expecting you to draw thousands, right? But reasonably expected for you to be able to draw maybe 3, right? 3 peptides, 3 amino acids joining together, so a tripeptide. Secondary structure is the intermolecular forces, so non-covalent, between the amide links, right? So we're not up to anything to do with the R groups yet. So we've got hydrogen bonding occurring between right? So if I had, say, this is part of the chain and this is another chain linking nearby, then the lone pair on the nitrogen would be attracted towards the C double bond O here, right? So there's one hydrogen bond, and we'd see another here, right? So it's that hydrogen bonding occurring between the nitrogen from one nearby amino acid chain, polypeptide, right, and a corresponding one. And it relates to these 2 types of shapes. One is helical, right, referred to alpha helices, or this bending occurring, which we call, right, folding or pleating, and that's referred to as beta sheets, right? And this works out when the R groups are in alternating directions. So they're not all facing up, but they're alternating, causing that folding to occur. The helices occur when the R groups are all facing outwards. Right? Tertiary structure, right, is the overall 3D shape, and this refers to, right, this is created by the type of bonding between the R groups, right? So let's build this back up. Primary, order of amino acids, covalent bonding. Secondary structures, hydrogen bonding between amide links. Tertiary structure, overall 3D shape, influenced by the type of bonding occurring between the R groups. And there's lots of different type of bonding that occurs. You can get ionic, right? And because when we look back at the zwitterion, we know that whether that amino or carboxyl functional group in the R is influenced by pH, right? So at high pH, that NH2, right, becomes an NH3, so it becomes a positive charge. Therefore, we can create ionic bonding to occur. Covalent bonding we can get. The only type of covalent bonding with the tertiary structure between R groups is between 2 cysteine residues. So even though methionine has got sulfur, it won't form disulfide bridges. So disulfide, di meaning 2, sulfide referring to sulfur. We get a covalent bond occurring between those 2 sulfurs, between 2 nearby cysteine residues. Hydrogen bonding can occur between OH, COOH, and H2 residues, the later in their non-ionic form, right? So when I've got that either neutral pH, yes, you get the average in the zwitterion, but the neutral pH remains as COOH or NH2, right, at pH 7. But pH dependent on whether it's going to be hydrogen bonding occurring or ionic. We get dipole-dipole between other polar functional groups, right? So when I've got that NH or N within a ring or something like that, and so on. And then obviously dispersion forces between non-polar functional groups. And we know that that's the weakest form of intermolecular forces. Any questions on that? Yeah?
Ionic is only between metallic and non-metallic.
No, ionic is between cation and anion, right? So mostly ionic bonding occurs between metal and a non-metal, but that's a simpler definition of ionic bonding. Ionic bonding really is anion and cation, positive and negative charges. But we're referring to absolute charges, so things that are charged, hence NH3 and COO minus, ionic bonding occur. And lastly, the quaternary, right, the fourth level of structure. And this is when I've got two or more polypeptide chains and some non-protein molecules might be involved, produces larger, more complex functional units. And the example there is hemoglobin, right? Hemoglobin's within red blood cells. You've got an ion at the center of hemoglobin that allows for the binding of oxygen to go through. So this is four separate polypeptide chains known as myoglobin that join together, right, in a complex shape to give us hemoglobin, the oxygen carrier within blood, right? And that links back to, right, equilibrium, yeah? So we've come sort of full circle. And remember that questions on the exam can be across multiple areas of study. So it's fairly reasonable that I can ask you a protein-related question on hemoglobin, right? And then have a subsequent question talking about how hemoglobin relates and why it binds to carbon monoxide more. Yeah? Right, so, and then right at the end of the unit, we talk about energy of food, right? So that links right back to the start of Unit 3. So it comes full circle. Okay. Draw and name the structure of the two amino acids produced when the dipeptide undergoes hydrolysis. So remember, condensation is the joining, hydrolysis is the breaking. How would you go about doing that? Get out your data books. Yeah, recognize the R groups within this dipeptide. Yeah, one of them's already on the board. Yeah, so we've got glycine. Yeah, glycine. And what's this one? CH2OH. Serine. So how we answer it, step one, find the amide functional group. I'm going to split that in the middle. Look at that. Yeah? So find where the amide functional group, I'm going to split that. We're going to add an OH to one side and an H to the NH side. We identify the R groups, we've done that. Yeah? Got an H on one side, right, which is our glycine residue, and we've got, right, our serine functional group on the other side. Match the R groups with the amino acids on the table, glycine and serine. Right, name and draw accordingly, as shown. All right, BCAA question. 2014. Amino acids can be classified according to the nature of their side chains, R groups. They may be polar, non-polar, acidic, or basic. Refer to the data book. Name one amino acid that has a non-polar side chain, one that has an acidic side chain. Right, non-polar side chain? Give me one. Alanine, go. Yep, alanine would be one non-polar side chain and an acidic side chain. There's only two. Glutamic acid or? Aspartic acid. No. Asparagine acid. Yes. No. Be careful. Yeah? Really surprised, right? Only 58% got 2 marks. Easy marks. Yeah? Amino acid with a non-polar side chain. Any one of alanine, glycine, isoleucine, leucine, methionine, See? Non-polar, methionine. All right, so I'll fix that up. Methionine, non-polar, despite the sulfur. Phenylalanine, proline, valine, or the three-letter abbreviation. Amino acid with an acidic side chain, one of aspartic or glutamic acid, ASP or GLU. Most students are able to identify the amino acid with a non-polar side chain. The acidic side chain proved to be more challenging. I don't see how. There's only two of them, and they've got acid in the name. Right? Really surprised. 13% of students got nothing. Right? That's one of you in this room that didn't get anything. It's either not answering the question, right? Okay. Table below provides examples of different categories of side chains at a pH of 7. Right, so it's showing you what the side chains are at a pH of 7 of these amino acids. So have a quick look. We'll come back to the chain. Come back to the table. Tertiary structure of protein is a result of bonding interactions between side chains of amino acid residues. Use the information provided in this table. 2. 1. Identify the amino acid that is involved in the formation of disulfide bonds. Cysteine. Boom. Got it in 1. 2. Give an example of two amino acid side chains that may form hydrogen bonds between each other. Ned. Asparagine and serine. Correct. Yeah. Give an example of an amino acid side chain that may form ionic bonds, salt bridges, between each other. Asparagine, no. Lysine, yes. And aspartic acid. Why? The ions. Yeah? Identify the type of bonding that exists between two side chains of two alanine residues. Dispersion only. CH3 residue, right? Non-polar. 89% got the first one. Cysteine. Only 39% got the next one. Yeah? So overall, students did not perform well in parts 2 to 4 of this question. Answers to parts 1 to 3 were obtained from the table supplied in the question. Most students struggled to identify the groups on the given side chain structures that could form hydrogen bonds, OH and NH2, in part B2, or that could form ionic bonds, COO minus and NH3 plus in part 3. Students should be aware that hydrogen bonds can occur between species which hydrogen is covalently bonded to oxygen or nitrogen atoms. Aspartic acid and lysine, 45% got the mark. 35% got the mark for the first one. Some students responded with covalent bonding. However, side chains are non-polar methyl groups. So the only covalent bonding that occurs between R groups is disulfide bridges between two cysteine residues. I'll leave it there for today. So 15.1, 2, 3, 4 review is all proteins. And then we'll talk about fats and lipids next class. It's really straightforward. They're only essential because they cannot be synthesized in your body. That's the only reason. Yeah? So therefore, must be part of your diet. The others, of the 9, so if we go back to essential amino acids, I'll write that in the presentation. I'll go back and I'll add that in. So our essential amino acids, so of these 9, must be a part of your diet, as they cannot be synthesized in your body. Cool? All right. Yes.
I have a somewhat chemistry question.
Yeah.